Chapter 18, Regulation of Gene Expression, is going to be our topic for this. This will be what you are expected to complete over the week off that we have for Thanksgiving. So please stay on top of this, and you should be golden. When we come back, we'll look at Chapter 19, and then we'll start breaking it down for our final exams. I'll probably be breaking this up into each section, so in this case we're going to be looking primarily at section 18.1. Before we get going, there's going to be a few questions I skip over. We're not going to be covering these 6 or 7 questions, so you can just go ahead and put a little X over those numbers. If it sounds like a topic that you're interested, by all means, go ahead. You can fill those in if you so choose, but I'm not going to be testing you on those ones. Number 45, we're not going to actually do number 45, but we are going to be talking about some very important information tied to that question. So when we get to that question, you're going to definitely want to pay attention to the information on the slide that I cover over that particular topic. Some quick review before we get going over the cell cycle and mitosis, as this will come up in reference as we go forward. The cell cycle has 4 stages: the G1, the S, the G2, and the M stage. During the G1 stage of the cell cycle, cells grow and they make a copy of their organelles. They go through a checkpoint to make sure that everything's in working order, and then they move to the synthesis stage. In synthesis, a cell makes a copy of its chromosomes. We're going to end up making 2 cells from the original cell that we started with. So we need to make sure that there's 2 copies of the DNA, one for each of the new cells. Go through a checkpoint, make sure that the DNA is appropriately copied, and then we proceed. Take note that there are checkpoints in each of these stages. At the tail end of this, 18.5, we talk about problems that might arise if those checkpoints are breached. And then G2, grow a little bit more, checkpoint, make sure everything's in working order, and then we enter mitosis or cell division. Mitosis consists of 4 phases: prophase, metaphase, anaphase, and telophase. Cytokinesis it works in conjunction with mitosis, but it's not a stage of mitosis. This is simply the definition is splitting the cytoplasm. It kind of occurs at the same time telophase does. But it's given its own little slice here. It's in blue because technically it's not part of mitosis, but it does happen during it. So concept 18.1 covers bacteria. Bacteria often respond to environmental change by regulating transcription. So breaking that down, it sounds like bacteria can alter the rate of transcription due to environmental changes. We're going to be looking at E. coli in this, bacteria. E. coli live in the digestive tracts of humans, so the large and small intestine. They do need nutrients. They're not autotrophs, they're heterotrophs. Where do they get their nutrients from? Well, you hopefully are putting 2 and 2 together. They're in our digestive tract. The food that we eat feeds them. E. coli needs the amino acid tryptophan, which we provide with the foods we eat. When we do not eat, E. coli turns on a gene to produce the amino acid TRP, tryptophan. This gene is called the TRP operon. You'll hear me say TRP. I'll say TRP, so TRP operon, and we'll be discussing that shortly. So to recap, E. coli can turn a gene on or off, so genes are not on all the time. So this explains why E. coli turns on or off genes based off the metabolic needs. Needs tryptophan, turns on a gene to produce tryptophan. When we're eating, turns that gene off. It can get it from us. We'll be using this image to answer questions number 2 and 3. So take a look at those 2 questions real quick. Pause. You can come back to this. Bacterial metabolism can be controlled in 2 ways. The first way is to adjust the activity of enzymes already present. In the left side of the image, we see feedback inhibition, which is a negative feedback process that shuts down the production of tryptophan. The second way, cells can adjust the production levels of certain enzymes. We see this on the right side of the image. Actually, there's several genes there: TRPE, TRPD, TRPC, TRPB, TRPA. And going down the middle, we'll see a metabolic pathway that we have seen previously. There's a precursor, and there's a gene that will produce enzyme 1, which can change the precursor into a different product. This is acted upon by a second enzyme, which changes it into a different product. Acted upon by a third enzyme, which changes it into a different product, and final product, in this case, tryptophan. Genes E right here and D right here are necessary to make enzyme 1. So these two genes are necessary for the production of that. We need gene C, TRPC, for enzyme 2, and we need B and A for enzyme 3. Regulation of expression is also negative feedback. So we can shut off these genes which will block the production of tryptophan depending on what's present, what's not present. So thinking back, if genes E and D are turned off, there's no enzyme 1. We cannot break apart our precursor into this product. If there's no B or A present, if those genes are turned off, we can't make this third enzyme, so we could not manufacture tryptophan from this. There would be no tool here to break this apart into tryptophan. Then going back to the left side, regulation of enzyme activity, we can see this is going through a process. Precursor is acted upon by enzyme 1, new thing acted upon by 2, new thing acted upon by 3, producing tryptophan, and the production of tryptophan will, negative feedback, be interpreted by the first enzyme, and it will shut this process down when there is enough tryptophan. So moving on, looking at operons, the basic concept, and we'll be talking about questions 4 through 7 at this point. Operons are kind of big deals. They are sections of DNA that control transcription. There's a couple types of operons that we're going to be talking about. We're going to be talking about the TRP operon used in the production of tryptophan, and then the LAC operon used in the production of lactose, which is a sugar. Operons, they have different sections. Two of those sections are operators and promoters. A promoter is a site where RNA polymerase can bind to, and it promotes things. So it sounds like the promoter is somewhere at the beginning of a gene. The operator is a section of DNA that is the on-off switch. Seems to make sense that an on-off switch would be located before the gene itself as well. So if we had a long strand of DNA, there might be a gene right in this little stretch right here, and then we'll have our promoter and operator occur before it. So right in this region, if you can envision a line there. And then down this last bullet talks about the 3 parts of the operon. It includes the promoter, includes the operator, the promoter, and then the genes that they control. Those would be the 3 parts for question 7. So continuing with the basics of operons, we're going to be talking about repressor proteins and regulatory genes, questions 8 and 9. Regulatory genes, they are located in the DNA away from the operon, and they have their own promoters. They are expressed continuously, but they often require a trigger to alter their state of being turned on or being turned off. The operator or the operon that we will be talking about right now will be the TRP operon, and you'll see a picture of that shortly, so don't freak out if you're trying to envision all this. Just kind of absorb it. Take down notes over these words, and then we'll apply them to an image in a couple slides here. Anyhow, the TRP operon, or TRP operon, it's a section of DNA responsible for the synthesis of the amino acid tryptophan. Because regulatory genes are always expressed, there is always a repressor protein present. However, the operon is not always turned off. This is because the repressor itself is reversible. This means that when tryptophan is present, the repressor switches to an active state and shuts off the trip operon. So repressors, when they're active, they turn off. And it seems to sort of make sense because when you repress something, you're pushing something away, turning something off. And that's exactly what's going on. We are turning off the production of tryptophan by shutting down the trip operon. However, when tryptophan is not present or low in quantity, we need it, so the repressor switches to an inactive state and the gene turns back on and we start producing more tryptophan. And by we, I'm talking E. coli. Humans and mammals do not have this operon. So once again, repressors, they can be active or inactive. They turn on or turn off operons. And this depends on the presence of certain items. If certain items are present, they will be turned off, and if certain things are not present, they'll be turned on, and they'll affect the production of things from the DNA transcription. A co-repressor is sometimes used. We'll be looking at a co-repressor shortly. And it's a molecule that kind of works in conjunction with the repressor protein to switch an operon off. Now looking back at the trip operon, the section of DNA that codes for tryptophan in E. coli, by default that trip operon is turned on. And the genes for tryptophan are synthesized. And then when there's enough tryptophan present, it binds to the trip repressor protein, turning the operon off. This looks exactly like what we saw back here with some feedback inhibition. When we produce enough tryptophan, it shuts the process down. No need to continue to create tryptophan. We already got enough. Now the repressor that shuts that process down, it's active only when there's enough tryptophan. Therefore, tryptophan would be a co-repressor. This is helping the repressor shut down. So putting all the stuff that we've been talking about just now into a visual format, here we go. We're looking at a trip operon, and you're going to want to label some of these words, specifically the ones that are in red, on the additional sheet that you've been provided. This was not part of the reading outline, although it should be. So this is an example of a repressible operon. The gene is active, but it can be repressed when tryptophan is present to activate the repressor protein. So what we see here is this entire section of DNA, which is the trip operon. So we can see within the trip operon there is a promoter region, and within the promoter region there's a section called the operator. And then we have the genes that code for the various enzymes of tryptophan. Each of these genes, they'll be expressed and they will create enzymes necessary for the production of the polypeptide of tryptophan, that amino acid. You'll also notice that in some cases promoters are located away from the operon itself. In such cases, promoters over here are regulatory genes. They're not anywhere near this operon, yet they still control the functionality of this operon. So there's a couple different ways that these things can be regulated, either by the initial promoter that's part of the operon itself, or by distance regulators called regulatory genes. At any rate, when the operator is active, meaning that the repressor is inactive, we can see this protein right here, which would bind nicely into that snug little fit there. It's not bound there, so this is an active operon. This allows RNA polymerase to bind to the promoter, and RNA polymerase will begin transcribing some mRNA for tryptophan. Now these distal regulatory genes away from the operon, they're always active, but right here they have no function. That's because this repressor, it needs its co-repressor, it needs tryptophan. So if tryptophan is present, now this promoter, this regulatory gene can, in conjunction with tryptophan, activate a repressor. A repressor will bind to the promoter, and this blockage here will prevent RNA polymerase from binding to the promoter to begin with, and then no transcription of tryptophan can be made.